Today I'm going to talk about two projects related to wildlife and nutrients. It's not really going to be so much about wading birds today, a mixture of wading birds and waterfowl. The first project I'm going to talk about relates to the surveys we're doing on wildlife in the stormwater treatment areas, the SDAs, that are just north of the Everglades. And the question we're trying to ask there is, how do these wildlife affect the sort of nutrient dynamics and the ability of these SDAs to remove phosphorus from the water? And the second project I'm going to talk about is our active marsh, a series of studies we're doing called active marsh improvement. And basically what we're trying to do there is trying to figure out these different management strategies for improving the nutrient-enriched regions of the Everglades for wildlife. So I'm going to start off with the SDA surveys. But before I get into the detail, are you hearing that echo? Is that just me? Before I get into the details, let me just give you a little bit of background on the SDAs and you know, just some justification for why we're doing these surveys. So, where's the little button thing? Is that? There we go. Okay, so we have 5 SDAs at the northern edge of the Everglades, totaling about 57,000 acres in treatment wetlands that were designed, built, and operated pretty much according to various state and federal mandates, not least the Everglades Forever Act of 1994. And their primary goal is to reduce this phosphorus that's from stormwater runoff before we discharge that water into the Everglades. And we have a water quality-based effluent limitation for that phosphorus concentration of 13 parts per billion. So before that water goes into the Everglades, it's got to reach that 13 parts per billion. Okay. So these SDAs are pretty amazing at actually reducing the phosphorus loads. As we can see from the 5 different SDAs here, the concentrations of water, phosphorus going into these SDAs in the brown versus what's coming out in the blue. But we're still not quite getting to that 13 part per billion target. And so as part of a recent consent order, the district became responsible for developing what's known as the Restoration Strategies Science Plan. And basically, that science plan is to try and figure out ways how we can reduce that phosphorus at the outflow to get us down to that 13 parts per billion. So one of the key studies that I'm involved with as part of this science plan is to figure out the sources, transformations, and movements of phosphorus in the SDAs, basically to try and figure out the phosphorus cycling, how the phosphorus cycles through that SDA environment. And in general, the way these SDAs function is actually quite simple in theory. Water flows into these SDAs laden with nutrients, particularly phosphorus. Then we have various different species of plants, such as cattail, submerged aquatic vegetation, and periphyton and other algae that absorb that phosphorus as part of their growth process. And then when those plants die, basically what happens is the dead material sinks and it gets buried in the soil sediments of these SDAs, and the outflow water is effectively much cleaner. Now, that's all right in theory, that's how it works, but we don't really understand the mechanisms and details of how that operates. And if we can improve our knowledge of that, the idea is that we can sort of figure out how to manage these SDAs better. And one component, notice the ibis here releasing something into the marsh. One component that we really don't understand is how wildlife affects this nutrient cycling. Now, wildlife may be particularly important in the SDAs because the SDAs support such a highly abundant and diverse community of animals. For example, we have very high densities of aquatic animals such as fishes and macroinvertebrates. And what seems to be particularly dominant in some of these SDAs are these exotic species, the blue tilapia. And by the way, these aren't little fish. I don't, if folks aren't aware of what blue tilapia is, this was taken from 100 feet in a helicopter. So these are this size, and that is not an isolated shoal of fish. That you can fly over it and you'll just see that continuously for a good mile as you're flying along. So there's an awful lot, and these are benthic feeding fish, and that's important to note for later on. Okay, so lots of aquatic species. You know, these STAs are highly productive because of all the nutrients, but we manage them. We keep them open, so there's a lot of varied different types of habitats, and they support about 200 species of birds, some of which are really abundant, such as these wading birds and white pelicans, and of course the waterfowl, the ducks and rails and coots, etc. And it probably has some of the highest densities of alligators, certainly the highest densities I've ever seen in Florida, outside an alligator farm. And I like to say that, you know, this picture tells the whole story, but really it doesn't, because these alligators, and they're all large alligators, by the way, at least 8-9 feet long, big, healthy, fully fed. This stretched off for another half a mile odd into the distance, and they were on this side of the bank as well. So there was about 1,000 gators, I estimated, in that one cell, in that one SDA, which is pretty amazing. So, you know, and that's a huge biomass of animals, particularly for a predatory animal. So just imagine how much food there is for them to feed on, those tilapia I was talking about. Okay, so given those animals that we have there, you know, the birds, the fish, and the alligators, we think there's probably 5 main mechanisms by which these animals affect nutrient cycling in the Everglades. And the first is simply by consumption of food and excreting it. And that's important because basically it fundamentally alters the natural material and the rates of nutrient cycling. So, when an animal eats something, it's basically taking that stored package of fish or plant, and it's making it into a soluble packet that gets into the water column. But, that's an issue in itself. But what we're really concerned about are those animals that forage within the sediment, such as those blue tilapia and armored catfish I was talking about. The issue here is when an animal feeds in the sediment, remember the SDAs, remember the way they function is they're storing that phosphorus within the soil sediments. So when you have an animal like this armored catfish feeding in the sediments, it's actually transporting that stored nutrients, that stored phosphorus, via eating and excretion back into the water column, and it's actually increasing the phosphorus levels in the water column. So consumption of food can also affect the nutrient cycling indirectly, particularly when the food source that they're eating are the very plants, the SAV, that the SDAs have planted and designed to actually remove the phosphorus. So some of these coots can be found in huge numbers, and I'll show you some data that we have a bit later on. Not just coots, we have ducks as well, but not so many numbers. But also exotic apple snails. We can have, in one SDA, we have literally millions and millions of exotic apple snails at certain times. And they have been known, them and the coots, to wipe out huge areas of these SAV beds, submerged aquatic vegetation beds. And so obviously when that gets removed, the SDA stops to function as a phosphorus removing process. Thirdly, these certain animals can affect the nutrient cycling through the effect of disturbing the sediments and actually, bringing that stored phosphorus back up into the water column, pretty much like the actual consumption I was talking about earlier, but this time just in the manner of how they forage, how they nest, how they move. So we have, these are, I don't know if you can see very easily, especially at the back, but this is an aerial photo of the wetlands, and basically dotted all in amongst here are thousands of fish nests from those blue tilapia and Mayan cichlids and other species. We have alligators here that are creating these trails and disturbing the sediments this way, that way. And then we have birds and other animals, these large American white pelicans here, creating this big flume of sediment here during their feeding process. Now, I've talked about the last three mechanisms have been about how animals might increase nutrient loads back into the water column. But certain animals might actually be able to reduce nutrient loads as well. So one of the ways they might be able to do this is acting as storage for phosphorus. So here we have these alligators. They're very large and long-lived. They live up to about 50 years, and they store huge amounts of phosphorus in their bones and their scutes, in their skin. And because they live such a long time, you're effectively removing the phosphorus by storing it in their bodies. Another way is that certain animals can actually transport phosphorus, maybe in the way of fish. A good example of that is these wading birds, which can feed in large numbers in the SDAs, remove the fish, and use it for a good purpose, which is to grow more wading birds. The STAs are close to one of the largest wading bird colonies in the Everglades. It's only a wading bird flight away, and that colony can have tens of thousands of birds. I see the light. So that's one way. We can actually potentially use these wading birds to get to as a tool to remove some of the phosphorus and actually have a benefit in terms of restoration as well. Have we not figured that out? I'll carry on. So, given all that, and the fact that we don't really know much about the wildlife in these SDAs, there's been very little research done on them. Our main goals are really just to get some really good population estimates for these species, particularly those species I was talking about, in this case the birds, the water birds, and some of the aquatic species, the fish and the macroinvertebrates. And then once we've done that, there's a lot of data already in the literature about their consumption and excretion rates, so we can actually do these population level calculations on how much they're excreting and consuming, and we can figure out what the actual effects are of these animals on the SDAs. So how are we doing some of these surveys? I'm going to focus on the birds just because of time limits. So the district is conducting basically their aerial video surveys of the SDAs. Basically what we do is we put a GoPro camera on the front of one of these helicopters, and we fly these fixed transects across the open water cells of these SDAs. The SDAs are comprised of two different types of large open water wetlands. They have basically cattail emergent cells, and then these more open water cells, and the open water cells obviously is where you're getting the majority of your birds. So we're flying over those open water cells, fixed transects, and they cover about 10% of the aerial coverage of those wetlands. And we do those to get a good temporal sense of how these birds are using these wetlands. We do it during the overwintering period from November to May, and we do it twice per month, so we can get a really good handle of how the birds are using these SDAs. And I wanted to actually do it in 5 of these SDAs, but it was so. The actual surveys themselves aren't labor intensive, but processing those videos, I have a video later that I hope I have time to show you, showing some of the birds, and you'll see why it's so labor intensive. So I've limited to 3 of those SDAs. Okay, so we're still at the process where we're analyzing those videos, and we're not quite there yet to actually do the calculations of the population estimates. But I do have some raw data that I think is very telling. So this shows the number of birds we counted in a single day in SDA 1 West in blue and SDA 2 in orange. And it's broken down into the different groups of birds. We have coots, dabbling ducks, diving ducks, whistling ducks, wading birds, and white pelicans. And what we can see is we have quite, when we add up all the different ducks, we have between about 4 and, wow, we have about 4 and 5, I counted about 4,000 or 5,000 ducks in each SDA, but it's really the coots that are incredible in terms of their numbers here. We got 14,000 in one SDA and over 12,000 in another. But what we have to remember here is this is a count from the transect area only. It's not a population estimate of the entire STA. So given we're only calculating 10%, we can probably, to get a very ballpark estimate of how many birds, we can probably times this by 10. So each of these STAs on that one day had between 120 and 140,000 coots and about 50 to 60,000 ducks. So that is quite an incredible number for a single day. And that's not just a fluke day that I picked out because I knew that there was a lot of birds there. Those numbers are fairly consistent every 2 weeks that I'm doing it. There's ups and downs, but that's a fairly average number of birds. In fact, I'll show you the video. We had over 100,000 ducks in one cell on one day. And so this is actually an underestimate to some level. So what kind of effects then do we think that we're having? So I can do a quick sort of back of the envelope calculation here and look at what some of these birds might be doing. So if we look at the coots, just using that very conservative estimate of 14,600 birds that we counted on that one day, and looking at some consumption and excretion rate data from the literature, we can do a calculation, and basically on that one day, these birds consumed 1,300 kilos of SAV and almost 800 and released 800 kilos of guano. So, and then remember, that's just the counted birds, so that's, if we extrapolate up to what we think that population is, that turns into 13,000 kilos and then of SAV and 8,000 kilos of guano, which is an incredible amount. I mean, and that's one day. Imagine that's going on all the winter, that they're consuming all that SAV. So that tells me, without really getting into the nitty-gritty of it all, that these birds are probably having a major effect on the nutrient cycling, because that, and these birds are doing it in two ways. That we're removing the SAV, so that's removing the process of removing the phosphorus, and also we're adding a whole ton of it back into the water column. I'm not going to go into the details of this. I just want to let you know that we are working on the fish stuff as well. So we've actually got a contract with Florida International University, and those guys are doing electrofishing and throw trap, which is a standard way of measuring fish in the Everglades. So those guys have started all that. I don't have any data for that just yet, but they're telling me that they don't have enough equipment to be able to deal with the amount of fish. These guys have been working in the Everglades for 20, 30 years, and they have a standard set protocol. They don't have enough equipment to deal with all the fish that they're collecting. So they've had to change their protocol. There's that many fish. Okay, so where is this leading us to? We'll have these great population estimates. It looks like there's going to be an effect. The next step, as we move forward over the next couple of years, will really be, once we've figured out which of those species are the key species that are affecting nutrient dynamics, probably some of those fish, probably those coots, then we can sort of really get into trying to understand what it is they like about the STAs, what type of SAV they like, what kind of water conditions. So maybe, and then with that, kind of figure out sort of potential ways of managing for those species. And when we're talking about managing, don't worry, we're not going to be getting rid of all the ducks or anything in the STAs. And a lot of it may be just sort of managing right by the outflows where we're measuring that sort of 13 parts per billion. So it's in that particular area that we really need to make sure that we're not getting that flush of nutrients back into the water column. Okay, so now I'm going to move on to the next study, and that's our active marsh improvement, which is looking at various different management approaches and various experiments we've been running to test how we can improve those habitats in the Everglades that have been just really ruined with excessive phosphorus inputs. So the issue here is, we've had decades of highly enriched nutrient-laden water, particularly phosphorus, entering the STAs, and that's converted, the traditional ridge and slough landscape. We've converted about 35,000 acres of that from our lovely open water sloughs and sawgrass ridges into this monotypic dead zone of cattail. And the area where that's really been most heavily impacted is this area called Water Conservation Area 2A. And this map here, the purple areas here are showing all the areas that are heavily enriched and covered in cattail. It's this area here that we're really working on and trying to figure out how we can improve it. So in terms of restoration, the problem is, even though we've really reduced the amount of phosphorus going into the Everglades these days through best management practices by farmers, and of course the SDAs that we've just been talking about, the problem is that the soil still is very highly enriched. And the reason that is because the cattail does such a phenomenal job of holding on to those nutrients and recycling it. Now this has two consequences in terms of how we want to think about restoring these systems. The first is that cattail is not going to go away anytime soon at all. It's probably going to be decades, if not over 100 years, before we see any sign of that cattail and nutrients disappearing naturally. It also means that cattail protects, by holding on to it, it protects downstream areas. So when we're thinking about management, we can't just go in and sort of nuke an entirely large, this huge area of cattail. What we need, otherwise we basically just end up with the nutrients flowing downstream, and we'd end up with a much larger area of cattail invasion. So, but there is one way, potentially, of breaking this resistance, and that's to create openings within the cattail matrix itself. And what we predict here is that if we create these openings, we would sort of will affect a change in the ecology and the food webs of these areas, these open water areas that will more closely match the natural Everglades ecology. So basically what we're trying to do here is sort of jumpstart restoration towards the sort of traditional Everglades ridge and slough landscape while we're waiting for those nutrient levels to decline. So we developed a study, a large ecosystem scale experiment in Water Conservation Area 2, where we tested whether creating these large 15-acre openings in the landscape, we could actually mimic the Everglades ridge and slough ecology. So we created, these openings were created in 2006 with a combination of herbicide and fire, and then subsequent herbicide applications thereafter as needed. And for each of these openings that we created, we also had a paired adjacent control that was just cattail. We just basically want to control what we're doing, what the difference is between opening these areas versus our controls, where we do no action. And we created 6 of these open control pairs. And we placed 3 of those pairs in a highly enriched part of Water Conservation Area 2A, and another 3 in what we call a transitional area. This is sort of moderately enriched and still, even though this area is totally dominated by, it's almost 100% cattail. This transitional area, it's got lower nutrients, and it's still got remnant sawgrass. There's still a lot of cattail. It's about 50-50 cattail to sawgrass, and it still has sort of remnant patterns of the ridge and slough landscape. We also created 3 reference plots because we wanted to compare our openings to the natural Everglades. So we had two main objectives. To test whether creating these openings within the vegetated areas sufficiently change the food web and the ecology and the wildlife usage of these areas such that we would increase wildlife use. And for that, we're comparing our open and control pairs, okay? For the second objective, we really wanted to say how these openings compare to the natural Everglades. So here we're comparing our openings to those reference sites in the natural Everglades that I was talking about. So I'm just going to show you some very brief results. We've been working on this 10 years. I could do a 2-3 hour talk on this, of all the results we have. But in a nutshell, pretty much as soon as we created these openings, after about a year or so, we got this really great growth of SAV, mainly Chara and periphyton, that created these really high oxygenated water level, dissolved oxygen levels in these areas. And that had an effect on the aquatic prey species in there. What we weren't expecting was that the cattail plots would have as much biomass of prey animals as the treated open plots. However, the difference is that whereas these cattail plots were dominated almost entirely by crayfish, which can pretty much live on anything, so they're living on the detritus of the cattail. It's dark in there, so there's no light, there's no oxygen, and they can deal with these very low oxygen levels. But our treated plots had a much more varied and diverse assemblage of aquatic animals, dominated by fish, but we also had crayfish, grass shrimp, the whole works, and it more closely matched that of the natural Everglades. Also of interest is the fact that these open plots produced a biomass that was 7 times greater than that of the natural Everglades. So we are really creating this sort of high productivity area for birds and other animals. So that's the basics of that. When we create these openings and we create all this extra food, we would predict that we'd get a lot more birds, foraging birds in there of all different types. And that is indeed what happened. So this graph shows, basically, it's the mean number of wading birds per week. So it's a very averaged out number of birds. So peak numbers are much, much higher than this, but this is averaged out over the entire dry season. So it shows the average number of birds per week for the first 9 years of the study. Yeah, and so this is great. We're looking at a really long-term, nice long-term study here. And we're looking at it in terms of the openings. So the enriched, the transitional area, and the natural unenriched Everglades. We're not looking at the cattail here because there simply weren't any birds feeding that dense cattail. So what we can see quite clearly is whereas there was really very little difference between the enriched and the transitional opening plots that we created, these openings that we created had much greater densities of birds than the natural Everglades. And what you're not really seeing from this graph is the way the birds used these plots. In the ridges and sloughs of the natural Everglades, it's a low nutrient system, and the way it works is that you really need that prey, the water levels come down, the prey to get concentrated, and the birds feed for a week or two in one place, and then they move off. With these openings that we created, the densities of prey were so much higher that the birds were able to feed there for months at a time. And not only that, the birds in the natural Everglades get very, they abandon their nests as soon as you get a rain event, your water levels come up, the prey disperse. These birds continue to feed in our open plots despite reversal events. So these open plots could actually function as a sort of mitigation against bad weather or improper water management that may affect the foraging birds, especially when they're nesting. And here we go. I'm not so interested in that. We'll skip that one. Sorry. He says PhDs don't have a sense of humor. All right, so ducks as well, waterfowl. We had huge numbers of waterfowl. Again, we're not really seeing the peaks in terms of the numbers of ducks. And in terms of ducks, it was mainly teal that we were seeing. Mottled ducks are in there as well, and the odd shoveler, but it was mainly blue-winged teal. What we found with the ducks is, unlike the coots and the wading birds that really didn't mind whether it was the transitional area or the highly enriched area, these ducks really did like the high nutrients. So if we want to create any of these openings in the landscape for duck hunting, then really we want to be doing it in those enriched areas rather than transitional areas. Yeah, and so the coots, no problem. And again, just like I said with the wading birds, the way they use these areas is pretty interesting. And what you're not really seeing there is the peaks in the number of birds. When you got the right conditions, when water levels came down, we could see 3,000, a flock of 3,000 ducks in just one of our little plots. We also have to remember that the Everglades ridge and slough landscape is not just openings, but it is the vegetation as well, the sawgrass and the dense vegetation. And so we wanted to look and see what sort of effect that the actual cattail has on the distribution of some birds that might be using it, such as some of these federally listed rail and rare rails and bitterns. And we found that the cattail, and it's probably because we had all the crayfish in there there in such high densities, but this habitat is really important for these species. We were getting an average of 10 and it's pretty much dominated by king rail. There was a few least bitterns and soarers, but it was really this rare, fairly rare king rail. And the numbers in the enriched region we were just hearing them all over the place. It was incredible. So this is another good reason why we don't want to just wipe out the cattail, because it does actually serve some good ecological function. Okay, so the study I just mentioned was called the Cattail Habitat Improvement Project. We went on to do another study. This time we created plots that were much larger. They're about 6 times larger than the chip plots. These are 93 acres. We created 2 of these. And the idea here is that we wanted to see whether we could recreate the ridge and slough landscape, not just create an opening, but see if we can really try and get to restoring the actual structure of the landscape. And we were able to do this because in 2011, our veg management folks found that using a herbicide called imazamox actually targets the cattail, but pretty much leaves the sawgrass and some of these other submerged aquatic vegetation species alone. And so we were able to spray an area and get rid of the cattail, but keep the remnant sawgrass ridges. And I think, as you can see, that it's not the perfect Everglades ridge and slough, but it really is approaching that. It's getting very close to that. And these plots actually had more birds, wading birds, per area of open water than our chip plots. And we think maybe that's because of edge effects and the way that obviously when you've got a huge area with all these edges all over the place, you've got a lot more edge effect. And there's some evidence that edges are very important for prey and how the birds are capturing their prey. So creating something with a lot of edge is actually very good in terms of wading birds. Now, this wasn't so important for ducks because we did this in the transitional region. So remember, the ducks prefer the highly enriched regions. So it did attract ducks, but not in the numbers that we were seeing in our chip plots. And we're doing it right now. In fact, today we are burning even more areas. We've got FWC that collaborates with us, the burn crew out in the Everglades, and they go out and they're actually burning some of our plots, hopefully today. We're trying to look, I haven't got any details of it up here, but basically what the next stage that we're looking at is, we're using herbicides here, and if possible, we don't want to use herbicides. We want to use the fire, as we were discussing just a minute ago. So we're looking at various different regimes of fire, herbicide, fire, to see whether we can minimize the amount of herbicides we're using. And so just in summary, in terms of some of the values, some of the benefits we're getting from this restoration approach, this actually is the graph from those large plots I've just showed you, showing there's an east and west plot, and it's the number of wading birds. So we're getting an average of 250 birds in there for 3 or 4 months at a time each year. So it really does attract huge numbers, huge flocks of birds, sometimes as many as 1,000 birds in there. So in summary, for both this the CHIP project and this second AMI project that we're doing. In summary, you know, it works. We're attracting hundreds of birds per week, both ducks and wading birds, which is, you know, a major part of Everglades restoration. What we didn't expect is that these plots are attracting birds when conditions are poor. So when we get these reversal events, we're still getting birds in there, as long as water levels are, you know, aren't so deep that the birds can't actually, you know, land in them. But, you know, conditions that affect birds in the natural Everglades don't seem to have the same effect in these plots, which is great. We know, you know, we've been doing it for 10 years so far, and unfortunately, I should show you a picture of what they look like now. They're covered in water lilies and Eliocharis grass coming out. You know, they're beautiful SAV everywhere. They really look great. And I think most importantly is they're applicable. This, you know, we can use that in many places in the Everglades and potentially beyond the Everglades as well. We've recently had some folks have just recently sprayed large areas of Lake Okeechobee using sort of similar sort of approaches. So, you know, it's applicable in many areas and many habitats. And I'd like to finish off just by thanking our collaborators, FWC, who I just mentioned. They've done a great job of burning these plots. We wouldn't have been able to do this study without those guys. And it's pretty amazing, actually, watching these large plots burn. They do such a great job. And I'd just like to mention some of my main co-investigators, especially Sue Newman, who I've been working with this project for the last 10 years. And how much time do I have left? I've got plenty of time. Okay, so I've actually got a video. I'm not sure if those folks at the back are going to be able to see it. This is just a video from the helicopter surveys, so it sort of shows some of the densities of birds that we have. We don't really need the volume if you want to turn it. It's just the helicopter noise. So. All right, so first of all, this is STA2, large numbers of snow egrets flying around, and they're all feeding on, this is all the SAV, the birds are feeding on, you know, these are mainly wading birds. What you're not being able to see is there are actually hundreds of blue-winged teals flying around in here as well, but they're so small that we're not picking them up. Again, more wading birds, white pelicans. You know, when there's a lot of SAV and the water levels are right, you can just have thousands and thousands of these birds. Again, this is STA2. So yeah, these are more wading birds. In amongst all these white wading birds, what you're not being able to see very well is that there's almost as many tricolored herons as well, but they're gray and you're not being able to see them so well. This is just it slowed down a little bit so you can see them. So these STAs really, you know, are very productive and really do attract a lot of birds. Again, this is STA 1 West. I'm sure some of you may have hunted in this STA. And I think you can understand why, you know, when I try to count, you know, these surveys are probably, they take about 3 hours to do all 3 SDAs. So I have 3 hours every 2 weeks of counting this. And yeah. It takes me a day to do about an hour of video analysis. Okay. Yeah, I want to show you the ducks. There's one part that I really do want to show you that shows just how many blue-winged teals that we can have. All right, so now here, I don't know, there's hundreds of blue-winged teals flying. That, you know, as you know, they're so small that it's hard to see. You know, these are wide-angle lens. You can see them all taking off. What I want to show you is when we get to this part over here, we're going to see that in a minute. It's going to fly around and then it's going to go to that area in a second. So I don't know if you can see, but these little dots are all the thousands of blue-winged teals flying. But you'll really see the incredible numbers that we can attract in these SDAs in the next scene. And first of all, it's going to show you in real time, and then it's going to slow down. Look for the birds taking off the water. That's when you can see them. See them going off?
Oh, wow.
See them all going off? And this was happening all over the place. And we flew back and did a brief count again, a very quick estimation, and it was somewhere in the, here we go, you see in slow motion. Just watch them come up here. Just bird after bird after bird after bird. All right, that's it. Any questions? Yeah, let's turn the lights on so I can.
I have a question.
Sure. No, we can't turn. Oh, there we go.
Years ago, I'm talking 30 years ago, so I went to a thing put on by Marty, one lister who's been mentioned before today. The Eric Livingston gave a presentation of these mini STAs, okay?
Right.
In other words, they were treating runoff from highways.
Okay.
And he said they're very effective until about 10 years down the road, and then they start becoming exporters of nutrients. You're telling me that the cattails capture, the submerged captures, and then lock it in the salt
Right.
sediments down below.
Right. That's the theory.
It's been there, what, about 10 years now, something?
Actually, longer than that, yeah. So I think SDA1 West has been there longer, yeah.
And you haven't seen any of that yet?
No, not right. No, that's an interesting point. I mean, I'm probably the wrong person to ask about that because I'm a wildlife ecologist and I'm really just come in to sort of figure out how the wildlife are affecting the nutrient dynamics. I'm not an expert at all on how the SDAs work and how they're storing, but that's part of that study that we're doing. The wildlife is part of a study looking at that nutrient fluxes and sources, and I think that might actually get to some of those questions about longevity of the functioning of the SDAs.
That's
Maybe.
the presentation you did last year here. Where they showed that you get to a point
Yeah.
where you build up enough detrital material.
Right.
Where you have to go back, you have to dry them out.
And you have to scrape them.
Remove the material.
Yeah.
And I
Right.
don't remember the time period, but there is a point where you're diminishing returns on removal of phosphorus.
That's interesting. Yeah.
You have plenty of time. Anyone else? Okay, thank you, Marco.
Oh, we got one.
Mark, go ahead.
Yeah.
I don't know if I misunderstood you, but you say that the birds add phosphorus into the water, or are the birds part of the cycle because they eat the fish or algae, whatever they eat from the water, and it's part of the biomass.
Right.
So it's a mass balance.
Well, no, it is a mass balance, but if that bird is releasing, so you're right, it could potentially be, and we've got to figure that out. One of the issues that we might face is that if they're pooping a lot by the outflows, then there's no chance for that poop to be reincorporated back. But also, we don't really know, it is a mass balance, but we don't know what the effects are, because we don't know how, you are changing fundamentally what that phosphorus becomes. It potentially becomes more labile and more soluble and more able to flow out of the system and then be picked up as part of that 13 part per billion, level. So.
But it's part of the phosphorus that is already in the biomass. It's not because the birds.
Oh, that's what you're asking. Well, I didn't mention that, but I did mention the fact that the birds can transport it out. There is a possibility that those whistling ducks that somebody liked to shoot, they actually feed in some of the farm fields too, so they potentially could be bringing extra nutrients in, and there's fish moving. We don't know a lot of how the animals, this would be a whole new study, how the animals are moving in and out of the SDAs. The fish are certainly moving through the canals and coming into the SDAs, so potentially, yes, they could be. It could be part of it. It's just, we can't do everything at once, so yeah, but good point. Oh, we got the gentleman over in the corner raised his hand first.
Mark, you've certainly piqued our interest on ultimately what are the mass balances to that question.
Right.
And one of the components I'm very curious and I'm hoping that you're resolving on is what is the productivity of the biomass in that system in mentioned the amount of biomass that the coots are consuming daily, and then phosphorus that's being excreted.
Right.
Ultimately, the uptake rates and the growth rates of that biomass probably
It's true.
dictate what is being
Right.
taken up, what is cycling out.
Right.
You know, so that
Absolutely.
My bias is I'm a plant guy.
Yeah,
so
yeah.
I'm very interested in how the plants are mediating.
Right.
Not only the cropping,
Right.
grazing, but then the resulting growth rates. It's very interesting.
Yeah, no, I agree, and that's why I didn't really want to get into what the effects are going to be, because we need to do further experiments looking at, you know, how the plants are responding. We've got some ideas to do some experiments where we have cages, and we're sort of kind of in the process of doing that right now, where we exclude the fish and the birds, and then have others where we allow just the birds in and just the fish in, and all sorts of things to see what the responses are to those, to the growth rates of the plants, because we just don't know what the effects are right now.